All right, everybody, we're going to talk today. What we would have talked about tonight, if you had me in person or online, is energy. And we're essentially going to be covering section 2.3 and 2.4 in the book. So again, like I said last week, remember that I don't talk about every single thing in the chapter. So please make sure you go back and read the chapter. Make sure that you have a full understanding of everything. And if you don't, feel free to send me an email. I have absolutely no problem meeting with you guys individually if I need to, or explaining stuff over email. I'm totally okay with that. All right, so 2.3 is kind of the end of what I consider to be the chemistry stuff. When we get to the macromolecules in 2.4, I start to feel like that's more biology, anatomy related. So in 2.3, again, the finishing up of the chemistry part, we first need to make sure that we understand what the term energy means. Okay, I briefly mentioned it last week, but energy is the capacity to do work. And it kind of makes sense if you think about it, because if you have lots of energy, you're able to do more work more effectively. So in a system, being your body, ecosystem, whatever, the more energy that's there within a molecule, within a body, the more work it's able to do. And then we define work as the ability to move something. Okay. Now, in the body, you need energy to do a lot of things. One of those is breaking bonds, which we'll talk about a little bit today. You need energy to build molecules. We'll also talk about that today. You use it for muscle contractions, you use it for pumping blood, use it for a lot of things. So having energy in your body is really important. Without it, we would not be able to survive. Now, there are a couple different types of chemical reactions. We're going to go over 5 in general, one of them sort of in-depthly. Okay, so now when I write out reactions, because I want to make sure that I can get through this and there's not a whole lot of dead space time, when I write RxNS, it's my shorthand for reactions. So the first type of reaction that can occur in the body or in general is called a synthesis reaction. Okay, synthesis is just a fancy term for making something. Okay, and in a synthesis reaction, essentially what you have is you take two molecules that we're going to call A and B, and you join them together. Okay, to make one molecule. Now, let me go ahead and also orient you to what a chemical equation looks like. Okay, usually on the left-hand side, you have the reactants, so the things that are needed for the reaction to take place. And on the left-hand side of the equation is going to be your products, the things that are made in the reaction. Now, the arrow in the middle is showing the direction of movement, the direction that the reaction is going. Okay, so if it was where the arrow was pointing to the A plus B, that would actually be a different type of reaction. Okay, the second type of reaction that we're going to talk about briefly is called an exchange reaction. Okay, so essentially what you have are two molecules that are going to have atoms that are going to switch places. Okay, and we can record that or write that down as molecule AB in the presence of molecule CD will react and come together and be molecule AC plus BD. Okay, so all they've done, all that's happened is the B and the C have exchanged locations. Okay, now reaction number 3 is called a decomposition reaction. Okay, and a decomposition reaction is the exact opposite of a synthesis reaction. So you start off with a compound of some kind, and what you're doing is you're breaking apart that compound. So you end up with just its individual parts. Okay. Reaction number 4 we call a reversible reaction. Okay, and so just like the name suggests, this reaction can go either way, and really it depends on what's available. So the one they give you in the book, and the one that's really the most abundant reversible reaction in your body, is this one right here. I'm actually going to give you real reaction, not just letters. So you have carbon dioxide in the presence of water. That's essentially what that plus sign means. It's in the presence of. Excuse me. Okay. In the presence of that, I'm doing these arrows just slightly different. These arrows like this, they mean it's reversible. Actually, I might need more space. It'll be all right. Okay. This is, when those two are together, they become what we refer to as carbonic acid. And it is written as H2CO3. That's supposed to be an O, not a 6. Okay. Which can then turn into HCO3, which is a bicarbonate ion. Okay, and it's negative because it has an extra electron. And again, I realize this is not written exactly correct because I'm running out of space, but you get the idea, hopefully. And you're going to have a hydrogen ion here. Okay, so again, the direction of a reversible chemical reaction depends on what's available. So if in the body at any point there is too much carbon dioxide, okay, the reaction is going to go in this direction. If at any point your body is too acidic, okay, too many hydrogen ions floating around, then the equation or the reaction will go the other way. So it depends on what's available, what substrates are around. Okay, now the fifth one, and this is the big one, what we refer to as metabolic reactions. Okay? And there are two types of metabolic reactions. Okay, one of them, I'm going to do this in kind of a little table, is called a catabolic reaction. Okay, just going to do a little bit of line here. And the second one is called an anabolic reaction. Okay, let's talk about catabolic first. So in a catabolic reaction, okay, what's happening is we are breaking bonds within a molecule. Okay, while we're breaking bonds, we have energy that is being released. Okay, and because energy is being released, energy in the form of heat, okay, we call it an exergonic reaction, which means it gives off heat. Okay. The catabolic reaction of breaking ATP, which causes your muscles to shiver, gives off heat, which is what warms you up when you're cold. Okay, anabolic reactions are exactly the opposite of catabolic reactions. Okay, usually they are building molecules. Okay, because we're building molecules, instead of releasing energy, we are taking that energy and storing it in that bond. So they refer to it as energy absorbing, because energy is stored in the chemical bonds of a molecule. And we can call it endergonic. And what that means is it absorbs heat. Okay. Which makes it feel cold. Okay. Now, the way I remember this, so last week I was talking about anions and cations, and I said when I think about cations, I remember a cat and how cat likes to hide things from their owners. So the owners lose things. Okay. I'm not really a cat person. They're cool. I don't have one, but for whatever reason, I use them a lot to remember things in chemistry. So catabolic, cats are destructive. You always see videos of them breaking and tearing things apart. So I think of catabolic being destructive, breaking bonds. They have tons of energy when they're, running their zoomies around the house. So cats, catabolic, break bonds, energy released, and they give off heat. Now, move on to the next slide here. There are two major types. So those were categories of metabolic reactions. So the two types of metabolic reactions always go hand in hand, and there's two names for them. So we're briefly going to describe them, and then I'm going to give you a mnemonic device to remember them. So we have oxidation, and we have reduction. Okay, let's start with oxidation first. So in an oxidation reaction, an oxidative reaction, there's a molecule giving up an electron. Okay. When you're giving up an electron, you are breaking a bond. Okay. Essentially, you're giving off hydrogen. Okay. So we give up an electron. This is going to release heat or release energy. Okay. And it's called oxidation. Most of the time, it is because it is let me rephrase that. It's called oxidation because most of the time, it is oxygen that is what we call the oxidizing agent, or causing the other organism. Sorry, I have biology on the brain. Causing the other molecule to give up an electron. It's the what I call the electron acceptor. Okay, it is what is going to actually accept the oxygen from the molecule that is giving up that electron. Because remember yesterday I was talking about briefly about water and how oxygen is a electron hog, and so oxygen likes to come in and take up as many electrons as it can so that it can fill up its outer shell. Okay, now in reduction, it's the opposite. Okay, so in reduction, the molecule is gaining an electron. So when one molecule has given up an electron, another one is going to take that electron. Okay, so molecule gets an electron. When that happens, energy is gained by the molecule that is, by the atom, a molecule, gotta sneeze, that is getting that electron. Okay, the molecule that is gaining the electron is called the reducing agent. Wait, I got that backwards. The one that donates. I'm sorry. So the reducing agent is the molecule that donates. Okay. And the oxidizing agent, let me just make sure I got this correct. The molecule that takes the electron. Okay, so oxygen is going to take the electron. All right, so now what you probably are sitting there thinking is that if one molecule gives up an electron, then there has to be another molecule that gets that electron. Okay, so wherever there's an oxidation reaction, there is also a reduction reaction. We call those, because they are always coupled, redox reactions. Okay, when something becomes oxidized, there is something else that becomes reduced. Okay, Now, way to remember this. I'm gonna do it here. Okay, so way to remember this is, and I'm gonna put this over here on the side here. Remember the phrase oil rig. Okay, so oxidation, I'm gonna abbreviate that, is losing. So in oxidation, something is losing an electron. And reduction, again, I'm going to abbreviate that, is gaining. Okay, so if you can remember oil rig, oxidation is losing, reduction is gaining. You'll never get those confused. I wish someone had taught me that when I was in college. I did not learn that until I was in grad school. All right, let's move on to 2.4, which is all going to be all our organic compounds. Okay, so again, I want to reiterate, make sure you go back and read these chapters. I'm not covering every single point in this in the sections of these books because some of them, like this one, can be a little bit long. All right, so in 2.4, we are discussing organic compounds. And it's not the lack of fertilizer that makes a compound organic. Organic has everything to do with the presence of carbon. Okay, so not every carbon-based compound is considered organic. Methane is not considered an organic compound. Methane is CH4. It's actually inorganic, but most compounds that contain carbon are going to be considered organic. Now, what is it about carbon that makes it great for building things like the macromolecules we're going to talk about here in just a minute? Okay, carbon itself has, I'll just do black, who cares? 2 electron shells. So these dots here are the electrons. And if you remember, I said that first energy level, which is what I just drew, can only hold 2 electrons. Okay. The next one, that second energy level, can hold 8, but carbon itself only has 6 electrons. Okay. So we have the 2 on that first shell, and then we have the other 4 on the outer shell. Okay. On its valence shell. So it has 4 valent electrons. Okay. So we can call carbon tetravalent. Okay, 4 valence electrons. Now, that outer shell can carry 4. Can carry 8. Carbon only has 4. So that means we have the ability to gain 4 more electrons, and we want to because carbon, again, they wants to be full, has the ability to gain 4 more electrons to become full. So what this means is that carbon has the ability to form bonds with 4 molecules at once. Okay, what you'll see as we get through these macromolecules is that carbon is always the central molecule within these macromolecules. Okay, and again, it's because it's tetravalent, it has the ability to form 4 bonds at once with other molecules. Okay. All right, so let's define macromolecule. We'll change colors. Maybe. Okay, so macromolecule, macro is a prefix that means big, is just a huge molecule. Okay, huge. Okay, now molecules are made up of monomers, okay, which is just a fancy word for single unit. Mono means one, mer means unit. Okay, excuse me. And then when you put multiple monomers together, they make a polymer. Okay, I always use the example, and I would show you if I had the capability to do video right now. But usually when I think about monomers and polymers, I think about a Mardi Gras beaded necklace. Okay, each individual bead is a monomer. Okay, it could stand alone by itself. But when you string them together into a necklace, you're forming a polymer. Okay, so monomers are single unit, the bead, and the polymer is many units, 2 or more. If it's 2 units, it's actually called a dimer, and then from then on it's polymer, but we'll just go with many units for the purposes of this. Okay, let's talk about how we make polymers, okay? when I'm in my classroom, I talk about how my walls are made of concrete brick, and I say that each individual brick is a monomer. When somebody built them and put the grout and whatever it is in the middle, that's making a monomer or a polymer, which is my wall. All right, so when we make polymers, okay, we go through a process called polymerization. Anytime you see a TION word in science, it's often a process. This is a process of building polymers. Now, when we build polymers, we go through a process called dehydration synthesis. Okay, so when something is dehydrated or you dehydrate something, you're pulling water out of it. So dehydration means to remove water. Synthesis means to build. Okay, so when we are building a polymer, we're taking out water in order to put those monomers together. Okay, and this is what it looks like. So you've got molecule number 1, molecule number 2. Okay, I'm just gonna draw this real quick and then I'm gonna add a couple things to it. Okay, now let me add in, okay, off of one of those molecules, you're going to have a carboxyl group. Actually, off of both of them, you're going to have a carboxyl group, which is an OH. Okay, the reason it says HO on number 2 is just because the O is actually what bonds to the carbon in the backbone structure of molecule number 2. Okay, and there's an oxygen between molecules 1 and 2 when it becomes a polymer. Now, we have to remove a water. So what we're going to do is we're going to take the carboxyl group off of one, okay, and the hydrogen off of the other. Removing that hydrogen from molecule number 2 is going to free up a bond with that oxygen, which it is going to want to bond to the, it's going to want to, bind to the bond that we have opened up when we took off the carboxyl group off of molecule number 1. So now we go through our reaction. Okay, we have our polymer here. One is now bound to number 2, and we now have a water. So we have removed a water from these molecules so they can bind together. But what if we want to break them apart? What if we want to separate molecules 1 and 2? Well, we're going to do exactly the opposite. And this is a process called hydrolysis. Some people call it hydrolysis. It's not incorrect. Hydro is a word that means water. Lysis means to break. Okay, so we're using water to break, and it is exactly the opposite. So we start with molecule number 1 bound to molecule number 2. Okay, and I think we end up with molecule number 1 all by itself and molecule 2 all by itself. All right, let me add in the water here. Oxygen. Okay, and so water is actually going to ionize itself, so it's going to separate into that OH and a separate H. Okay, they are going to move into this reaction. Okay, and it's going to cause the bond there in the middle that just had a brain fart. The bond where they share electrons, covalent bond. There we go, got it. The covalent bond is going to break, and then the water parts, the ionized water, the OH and the H, are going to fill in into the gaps, and it'll go back to normal. So that is how we do that. Now, hydrolysis, I do want to add, and really both of these are accomplished with enzymes. And we will spend a little bit of time at the end of this talking about enzymes. Okay, there's an enzyme that's job is to break or build stuff. We'll talk about that a little bit later. All right, so let's talk about the first macromolecule. Carbohydrates. Okay, sugar, starch, stuff like that. All right, so carbohydrates have a general chemical formula of CH2O with an N. And what this essentially means, the simplest way, is that for every 2 hydrogen, there is 1 oxygen. So the hydrogen to oxygen ratio is 2 to 1. So for every 2 every 2 hydrogens, there is 1 oxygen. And I'll show you drawings here in just a minute. Now, carbohydrates are hydrophilic. Okay, this is we learned on Tuesday. It means that they love water, so they break apart relatively easy in water. Okay, and if you look, actually, I'll just draw it out here in just a second. Carbohydrates actually look different whether they are in the presence of water or not. So if they are not in the presence of water, they will form a long chain. So I'm going to draw glucose real quick in chain form and also in ring form. So in the presence of water, without water, I'm sorry, not in the presence of water, glucose will look like this. 1, 2, 3, 4, 5, 6. Double bond. Okay, you guys don't actually need to memorize this. Any ring structure is going to be a monomer. Glucose usually, sometimes fructose. Okay, so this is what glucose looks like if it's not in the presence of water. Okay, once you put glucose into water, it rings up and it will look something like this. Now, every single one of these, with the exception of one, every single corner here, junction, that's a carbon. That indicates a carbon. The exception is this one right here, and it's actually an oxygen. Okay. But I'm going to finish this out. Here you've got CH2OH. Actually, I don't want to do that. I want to do that. Okay, so both of these structures are glucose. Glucose is C6H12O6. Once we get to, I believe we cover photosynthesis, or at least cellular respiration. We don't need photosynthesis here. When we get to cellular respiration, it looks like we're going to do that February 4th. We'll talk a lot about glucose. So C6H12O6, don't forget that. You'll need to know it. Okay, that's what it looks like, either with or without water in the presence. Now, some examples of carbohydrates that you find in the body and in nature. So if there's only one ring, okay, it's called a monomer, one single ring. Examples of those are glucose and fructose. The fancy word is a monosaccharide. Saccharide is a fancy word for sweet. It's actually Latin, I believe. So glucose and fructose are both one single ring, like I drew on the board here just a second ago. The difference is just the arrangement of carbons and hydrogens and oxygens on there. If you have 2 monomers together, it's called a disaccharide. So you have sucrose and lactose. So lactose is the sugar that we can't digest if you are lactose intolerant. An important energy storage molecule for your body is glycogen. So animals will store their extra carbohydrates, their extra glucose as glycogen. This is stored in the liver, brain, the nerves use it a lot, muscles use it. You actually also find it in the vagina of females. For plants, they store their energy as starch. So the starch that we eat in potatoes, for example, is the energy store that plant has made. And they will use that energy when photosynthesis can't take place. So in the winter, when the sun is not as strong, or at night when the sun is not out at all. And then there's cellulose. So cellulose is another carbohydrate found in plants. and its job is to strengthen the cell wall. It is not digestible from us, or we are not able to digest it. Starch is actually the only large starch molecule or sugar molecule that we can digest. We can digest starch. We can't digest cellulose. Instead of digesting it, we still need it because what it does, it serves what we call fiber in a lot of common talk with medical, at the pharmacy and stuff. It helps to kind of clean our systems out because they swell when they come in contact with water. One way I have my students, my high school students, remember cellulose is when I tell them about when you eat corn. If you pay attention when you have a bowel movement, the next day, if you look down, you will often see at least the holes of the corn that you have eaten. The reason it does not digest and it looks the same sometimes as when you ate it is because your body can't digest it. Okay. Now, I do want to point out that carbohydrates will always end in OSE, glucose, fructose, sucrose, lactose. Now, these are the smaller ones. Obviously, glycogen, starch don't end in that. Cellulose is one. Okay, so that OSE, when you see a word that ends in OSE, that indicates that it is a carbohydrate. Okay, so they are mostly for energy, but there are a couple other ones that don't serve as energy, and they actually do things like signal between cells. Signal the immune system, kind of helps with cell identification. When we get to the cell membrane, what week is that? Cell membrane is January 28th, so in a couple weeks, we'll talk about the cell membrane. We'll talk about how there's carbohydrate chains attached to proteins and lipids and things, and we'll go over that. Okay, now, so if you remember, you only find, let me actually add that to it. You only find carbon, hydrogen, and oxygen in a carbohydrate. Okay, for lipids, let's do brown this time. That's definitely not brown. Oh well. Okay, you also find only carbon, hydrogen, and oxygen. Now I put those commas there because I don't want you to think that it is one carbon to one hydrogen to one oxygen. It is not. Okay. If you see a molecule that has a bunch of just carbon, hydrogen, and oxygen, just count the number and look at the ratio. Carbohydrates are in a 2-to-1 ratio with hydrogen to oxygen. Lipids are not. Okay. They have a ton of hydrogen. Okay. Now, lipids, as we know, anytime you try to mix an oil, for example, with water, it does not mix. So lipids are hydrophobic. Okay. And there are many types of lipids, okay? And they all, not all, but they serve many different functions. So we're going to go through those briefly. So one type of lipid that we talk about is a fatty acid. Okay. And there are two types of fatty acids that you see if you look on your the nutrition label on your food. We have a saturated and an unsaturated. Okay. Saturated fatty acids, the short way to put it is that they are chock full of hydrogens. You cannot add any more hydrogen. Okay, and they will look, I'm just going to kind of briefly draw something like this. Okay, there is no possible way that we can add any more hydrogens to that. Carbons can only form 4 bonds. All 4 of their bonds are taken on here. The other, so it's fully saturated, it has tons of hydrogen. Okay, the other type is unsaturated. And as the name suggests, and as you probably figured out, that means it is not full of hydrogen. Okay. Okay, and they often will look similar to the unsaturated with one big difference. So 1, 2, 3, 4. Okay, with the big difference is there's a double bond in there. Okay. Actually, let me. Boop. Okay, so as you can see, we could break that one of those double bonds between those two carbons in the middle, and we can add two more hydrogens onto this molecule. Now, not really related to the class, but what I've actually drawn here is a trans fat. So you look at the food labels and you see saturated fat and unsaturated fat, and under unsaturated fat, you'll see a thing for trans fat. In a trans fat, the hydrogens around the double bond of carbon are on opposite sides of the molecule. The other one, if they're on the same side, it's called a cis fat. So cis meaning same. Okay, just fun fact. Okay, fatty acids are used for energy. If you can read that. There's another one used for energy, and these are called triglycerides. You've probably heard of these. You get them all the time when you go to the doctor and get blood work. High density versus low density lipids. Usually these are triglycerides. So a triglyceride is a molecule that has what we call a glycerol head. That's this box that I just drew. And it's got 3 fatty acid tails on it. Okay. That's another one that is used for energy. I thought I had something about oil written on here, but I guess I didn't. Sometimes the triglycerides are made with saturated fatty acids, and they're straight like the one I just drew here. Okay, these types of fats are solid at room temperature, so things like butter, lard. Okay, and then you've got, I'll draw a different color, unsaturated fatty acids. Or I'm sorry, unsaturated triglycerides. So they have 2 saturated fatty acids. And then you have this third one. Sometimes I think there might be more than one that is unsaturated. We know it's unsaturated because the double bond between the carbons actually causes the chain to kink a little bit. Okay, the ones that have a kink in it like this, these are liquid at room temperature, so things like oils or plant-based fats, which is pretty neat. All right, moving on. Another type of lipid that you find in the body are phospholipids. If I spelled that right, hang on. Phospholipids. Okay, these look somewhat similar to triglycerides, except without the glycerol head, without the glycerol backbone, they've got a phosphate head, and they've got 2 fatty acid tails, like so. Okay, they make up a major component of your cell membrane. We'll talk more about these on the 28th when we talk about the cell membrane. The phosphate head is hydrophilic, so it loves water. The fatty tails there are hydrophobic, and so it dictates how the cell membrane is held together. So phospholipids, part of the cell membrane. Another type of fat are eicosanoids. Oops, that's supposed to be an E. Sometimes if you don't pull your pen far enough away from the smartboard, it doesn't think that you picked it up. Okay, eicosanoids are fats that serve as hormone-like chemicals that signal cells to do different things. So they're important for things like blood clotting, childbirth, things like that. So it's another type of fat. And the last type, I think it's the last type. Last type we'll talk about are steroids. Okay, steroids are, we all know what they are. They help with, growth. They help with sexual maturity. They're signalers in the body, but they all stem from cholesterol. So cholesterol has gotten a bad rap for a while. Your body makes it on its own. It's not something you need to get from your food, but you can. You can have too much of it, but your body does a pretty good job of getting rid of it. All right, and there's no special ending for lipid names. It just lipids. All right, protein. Now this is a big one. Proteins are some of the most diverse molecules in your body. So we're going to spend a little bit of time talking about protein. All right, protein. These are made of amino acids. So the monomer of a protein is an amino acid. I don't think I explicitly said it with lipids, but the monomer of a lipid is a glycerol and a fatty acid. They come together to make other things, but really it's the fatty acid is the monomer for the lipid. All right, so proteins are made of amino acids. There are, in all of life, 20 of them. So the same 20 that we have in our body is the same 20 that's in your dog's body. It's the same 20 that's in your plant. It's the same 20 that's in your fish. Okay. What makes everything different, all the enzymes, all the proteins for your body, is the order and what amino acids are there. So there's an infinite number of combinations for these proteins, and including the length of that protein chain. Now, there's a basic structure for protein. Okay, and it looks like this. Again, it has a central carbon. Okay, off of one side, you have an amine group. Here's where they get the name amino acid. Okay, and then there's the other group over here. OH. Okay, now every single amino acid has this on top. This carbon, central carbon, this amine group, and this carboxyl group over here. What makes every amino acid, all 20 of those amino acids, different is what they call this R, or functional group, that hangs off the bottom. Okay, it is the R group, that functional group, that makes every amino acid different, gives it different properties. Is it hydrophilic or hydrophobic? Is it positive or negatively charged? And all of those things will play into how a protein is folded. Now, before we get to the structure of a protein in general, okay, when we have two or more amino acids together, it's called a peptide. Okay, and amino acids are held together with a peptide bond. This is the only time in the body you're going to see a peptide bond. It's to hold together proteins. Okay, give protein structure. Now, let's get a little more in depth into protein structure. Okay, so there are 4 levels of protein structure. Okay, first level is primary structure. Okay, so this is the order of the amino acids. Is there a glycine next to methionine or something like that? It is the order of the beads on the string. Okay. Secondary structure. Oh, let me add this. It is held together with peptide bonds, as I just said a minute ago. Okay, but the peptide bond is what dictates and holds together the protein at its primary structure level. All right, second level of structure. Whoops, I didn't want to do that. Here we go. Is secondary. They didn't get super creative with their levels of structure, which is good. Less to remember. Okay, these are coils and folds. Okay, and there are 2 types, the 2 major ones. Okay, what we call alpha helices, or alpha helix. Okay, and again, it looks like a spring. So it's when the protein primary structure kind of coils up on itself. Okay, I shouldn't say there's what we call beta pleated sheets, or beta pleats. Okay, and this really is that primary structure bending like a pleat on pants. Okay, these are held together and determined by hydrogen bonds. Okay, so peptide bonds primary, hydrogen bonds are secondary. Okay, third level, surprise, surprise, is called tertiary. Okay, this is further bending and folding of that polypeptide chain that we originally built. Okay, and this essentially makes it turn into a globular shape. Okay, this is again, it's still more folding. It looks like a glob when it's finished. Okay, it's caused by a couple of things. Let me change this to pink. Okay, it is caused by, really, it's those R groups that are on the ends or hanging off of amino acids. So it's hydrophilic versus hydrophobic. So when a protein is being assembled and it's getting its tertiary structure, the hydrophobic amino acids will congregate to, or go towards the middle of the inside of the glob, and the hydrophilic ones will be on the outside because they're going to be the ones that come in contact with the water. Okay, but really it's the properties of the R group. Are they positive or negative? Okay, so the negative ones will be attracted to the positive ones, but it's all about the R groups. It's also so the properties, it is also going to be the van der Waals forces we talked about on Tuesday. We'll spell it wrong again. So again, those van der Waals forces are going to be the temporary attractions depending on where around the molecule the electrons are concentrated. And there's one new type of bond that forms only between the amino acids that have sulfur, sulfides hanging in their R groups. And we call these disulfide bridges. Okay. Now, after tertiary structure, I apologize having to go to a second page. We have one more level and then we're done. So quaternary. Okay, in quaternary structure, you have two or more polypeptide chains combining. So primary, secondary, and tertiary structure deal with one single polypeptide chain. Okay, when we get to quaternary structure, we have two or more polypeptide chains coming together. Okay, now an example of this type of protein, not every protein has a quaternary structure. Okay, some don't, they don't need it. But hemoglobin is one of the examples of this. Hemoglobin is actually a protein that contains 4 different polypeptide chains that have been folded and globbed together. And it has an iron stuck in the middle. Okay, and that's going to hold on to the oxygen in your blood. We'll talk about that when we get to blood. Okay. Now, proteins are pretty picky about the type of environment they're working in, okay? And they can change form based on what molecules are around and what the environment is like. Okay? And they go through what we call conformational change. Okay, conformational change is a fancy word for shape change. Okay, proteins at some point usually change their shape. Now, it's not always a bad thing. Okay, sometimes it's needed to perform a function. Okay, so we'll talk about enzymes in a minute. Sometimes that enzyme needs to actually fold around the substrate it's acting on in order to do its job. Okay, but sometimes too much movement is bad. Too much shape change, too much conformational change is bad. Okay, too much, we call that denature. Okay, if the shape changes too much, it goes through denaturation and it can't do its job anymore. And usually that is caused by too much of a change in pH, okay, or too much of a change in temperature. Okay. For example, in your digestive system, you have pepsin, which is an enzyme that works only in your stomach, and your stomach has a pH of about 2. Okay. Salivary amylase is an enzyme that works in your mouth, in your saliva, breaks down the carbohydrates before it gets in your stomach. As you swallow your food, salivary amylase is still in what they call the bolus that goes down into your stomach. But when it gets into the stomach and it comes in contact with the stomach acid, it denatures that amylase protein and it no longer works. Okay, so that's what happens. Now, temperature also, all of the enzymes in our body, they all have different pH ranges, okay, but they all operate at about the same temperature, about 37 degrees Celsius. It's one of the reasons why fevers are so dangerous. If you get above a certain temperature, the proteins in your body stop doing their job, which means you have a hard time surviving. All right, now let's wrap this up here pretty soon. So let's talk about enzymes. They are a type of protein. Okay, not all proteins are enzymes, but all enzymes are proteins. Okay, I'll say that again. Not all proteins are enzymes. All enzymes are proteins. Okay, collagen is a protein that is not an enzyme. Okay, now enzymes, when you see their name, they will always end in ASE. Okay, so lactose is a sugar. Lactase is the enzyme that breaks down the lactose sugar. Okay, they are named based on what they break down for the most part. Okay, now enzymes, we define them as biological catalysts. Okay, and what their job is to lower what we call the activation energy needed for a reaction to take place. Okay, now I'm going to draw a little figure for you. Okay, we're on the y-axis here. It is energy. On your x-axis is time. Okay, let's say that we are breaking apart something. Okay, so we're going to start with more energy than we end with because we're releasing energy. Okay, when I need to complete that, I need to kind of a jump start to get it going. Okay, so the amount of energy needed is going to go up. It's going to break down, and then there's going to be less energy in my product. Okay, this might be a reaction without an enzyme. Okay, remember that energy equals heat. Okay, if you're breaking something down in your body, you don't have the enzyme for it, you can still do it. It's just going to generate a lot of heat, and it might not be good for you. So you have enzymes in your body. And I'm going to do this next one in red. Okay, same chemical reaction. We're breaking down the same product, but now instead of going all the way up that hill, we have a smaller hill to go up. Okay, so it lowers the energy needed to get that reaction going. Okay, you generate less heat. Your body doesn't overheat. They give an example about burning paper. When you burn paper, you're burning glucose. That could happen in your body without enzymes, and that would be really bad. I don't want any part of that. Okay, so enzymes act on a substrate. Again, that substrate is usually what the enzyme is named after. So here in a little bit, just a few minutes, I'm going to give you a couple enzyme names, and I'll talk about them. But they're named after what they do, what they break down. Some enzymes are what we call take-apart enzymes. So their job is to break things apart. Okay, go through the decomposition reaction, breaking down larger molecules. Other enzymes are what we call put-together enzymes or synthesis enzymes, and they actually build things. Okay. All right, so when an enzyme works, okay, I'm going to do one of my amazing art drawings. Okay, you have a glob. Let's do this. Okay, there's my globular protein. It's beautiful, isn't it? Okay, if you notice, there's a little chunk taken out of the top there. Okay, that chunk is perfectly made for the substrate that needs to fit into it. Okay, something we call specificity. Okay, enzymes are specific on what they work on. The enzyme that breaks down ATP is not the same enzyme that's going to break down lactase sugar. Okay, they all have different enzymes in digestion. I keep going back to that because I'm teaching that now in my high school class. Amylase breaks down starch. Pepsin and trypsin both break down protein. Lipase breaks down lipids. Okay, so there's an enzyme for everything. Okay, when the substrate goes into the enzyme. So it goes into the enzyme. This part here, that V that I'm doing in orange here, so we call the active site. That's where all the magic happens. Okay, when it goes into there and it ends up being called, now I'm not gonna be able to draw this exactly like I did before, but we'll go with it. Okay, that's supposed to be the same enzyme. I know, it is beautiful. Okay, when it actually goes into the enzyme and goes into the active site, we call that the enzyme substrate complex. Okay, the enzymatic reaction takes place, and then what is released is your product. Oops, I meant to draw that in purple. Okay, again, supposed to be the same enzyme, should be the same shape, but I'm not good at drawing the same thing twice. And what you might end up with, what leaves, is a different product than what actually went in. Okay, that's your enzymes. Okay, enzymes will either hydrolyze or break or synthesize, make the substrate and release the product. That's what we just saw here. Now, the cool thing about enzymes is they are not used up in a reaction. Okay, so this reaction that I drew on the board here, the bringing in of the green triangle and the shooting out of the orange and the red and the pink triangle, that same enzyme will do it over and over and over and over and over and over again until the substrate is used up. Okay, if you put a potato in hydrogen peroxide, if you have it at home, you should try it. It's kind of fun. Take a little chunk of potato, put it in a cup of hydrogen peroxide, you'll see bubbles forming. Okay, inside potatoes and even inside your cells, you can do this with liver as well. There's a enzyme called catalase. Catalase breaks down hydrogen peroxide into oxygen and water, which is why it bubbles. Okay, if you have your potato, you pour out the hydrogen peroxide and you put new hydrogen peroxide in, it will keep bubbling and it will keep bubbling. You pour the hydrogen peroxide out, add more in, and it keeps bubbling. But if you have the potato and the hydrogen peroxide, it stops bubbling, and you take the potato out and you put a new potato in, it's not going to bubble because the enzyme is in the potato and you have used the substrate that is the hydrogen peroxide. The substrate's gone. The enzyme can't do its job anymore. Okay. And again, just like proteins do, enzymes will denature in the wrong pH or temperature. Again, that's why fevers are dangerous. That's why you want to make sure the pH of your body and your blood pH is normal, around 7. Otherwise, things won't work. Now, sometimes enzymes can be turned on and turned off. There are molecules called cofactors that will actually bind to an enzyme when it's time to work, and it will turn them on like a light switch. And when they're done, not needed, that cofactor will go away, the enzyme will turn off, and it won't need to work again. Enzymes are cool, I think. Can't survive without them. All right, last thing, I'll be done. A few more minutes. Thanks for bearing with me. The last type of macromolecule is a nucleic acid. Okay, we will talk more about DNA and RNA in a couple of weeks. But I would read through 4.1. It's super short. It just compares the two. If you have questions, ask me. But I wanted to kind of be short on time. Now, the base structure of a nucleotide looks something like this. You've got a phosphate group attached to some kind of sugar, and that sugar is attached to a nitrogen base. Okay. That nitrogen base can differ depending on what nucleotide you are in DNA. If you are an ATP versus a GTP, it's a little bit different. Okay. Now, ATP is the most important, some would argue the most important molecule in your body. It is what powers your body. Okay. Without it, you would not survive. Okay. And it looks somewhat like the base, with just a little bit of a different formulation here. So let me draw it for you. Now, ATP stands for adenosine triphosphate. Okay, adenosine is named for the adenine. Okay, that's ADENINE that I put there. That nitrogen base is adenine. It has a ribose sugar. It's just a type of sugar that's there. Okay, and 3 phosphates. That's where the triphosphate comes from. Okay, this is what we call the high energy form of this molecule. Okay, it is the most important energy molecule. Tons of energy stored in this bond right here, the one I just colored purple. Tons. These phosphates are negatively charged, so keeping them together requires a lot of energy. So there's a lot of energy stored there. Now, in the cell, when your body uses ATP for everything, including moving muscles, okay, this bond is broken. Okay, and it is broken by an enzyme called ATP ACE. Okay, or adenosine triphosphatase, but we'll go with ATPase because it's a lot easier to remember. Okay, it's named ASE, means it's an enzyme, and it's named for what it breaks down or what it does. So it breaks down ATP. Okay, and it breaks ATP down into ADP, adenosine diphosphate, plus an extra phosphate, just kind of hanging out there, getting ready to get put back on somewhere else. Okay. I like to refer to the relationship between ATP and ADP as a gift card. Okay. ATP is that full gift card ready to be spent. ADP is the spent empty gift card that can be recharged. You recharge it by adding that phosphate back. That phosphate is added back during cellular respiration by an enzyme called ATP synthase. Okay, so this is an enzyme that synthesizes ATP. Okay, now ATP is very short-lived. Okay, you're constantly making it. I actually didn't know this until I was reading up on this before I taught you guys this stuff. But in order for you to survive every day, your body needs to make 99 pounds of ATP. It gives you an idea of how important it is. We don't gain 100 pounds every day, so it's made and used like that. One of the reasons that cyanide, for example, is so dangerous is because it prevents ATP from being made. And if you stop making ATP, you'll be dead in about a minute. Okay, it doesn't take very long at all. Okay, so if you guys have any questions about anything we talked about, send me an email, let me know, and I will answer it as best I can. Have a good evening, guys. I will see you next week on the 19th online. We'll be back in person, hopefully, on the 21st, back in the building at Fort Knox. Have a good evening, guys.